Click for IB Biology. Here we're taking a look at the lysogenic cycle of the virus. There's a blank PDF of this presentation down in the description that you can use to annotate with your own notes. Then you can take the quiz to check your understanding. I've provided a prompt structure so that you can research this using AI. And then in the end screen, we'll link you through to the next video in the sequence, looking at virus diversity, to the virus playlist, and indeed, of course, to the channel. The lysogenic cycle. As with the previous video on the lytic cycle, the diagrams here are not to scale. I've also illustrated the virus with a simple capsid and nucleic acid image rather than the complexity of potential structures. So the virus here will then attach to the surface of the host cell and introduce the nucleic acid into the cytoplasm. The nucleic acid, depending upon when it's DNA or RNA, will then be replicated using the host DNA-RNA replication mechanism. With retroviruses, RNA viruses, these will be transformed into copied DNA by the action of reverse transcriptase enzyme, an enzyme which is actually encoded on the virus genome. So here at this intermediary stage of the infection, the nucleic acid now has been replicated many times. It's necessary for the DNA to become double-stranded DNA and take the form of a helix. This is then incorporated into the host chromosome and using the enzyme integrase. Integrase will cut the host DNA, and then it will seal in the DNA so that the chromosome is complete. There is some indication that the incorporation point of the viral nucleic acid corresponds to areas where genes are very active. At this point, it's possible that the virus will become dormant and will remain within the host DNA for prolonged periods of time. As the host cell replicates, one cell giving two cells and so on, then what happens is that the actual viral DNA, the viral nucleic acid, is also replicated. Later, it may be that the cell will enter the lytic cycle, allowing the virus to then go on to infect either other tissues or other individuals. It has been noted in the study of chromosomes of host cells that there appears to be DNA incorporated, integrated into the host DNA that never seems to break out of the dormancy. This is referred to as ERV, or Endogenous Retrovirus, this may remain permanently within the genome of the host cells. And this has been studied in many organisms, many different types of organisms, where the presence of these viral components indicates tremendously old ancestry to the particular viruses, in some cases. Appearing on your screens now are links to a brief video on the diversity of viruses, a link to the channel, and also to the playlist on viruses so that you can look at the rest of the series.